Good morning to you all. Today we continue with parasitology. Entamoeba gingivalis, which is the only Entamoeba species that is in the mouth, not in GI tract. It's in the mouth. That's why they call it gingivalis, from the gingiva. You know the gingiva? It's the tissue around your teeth. This is we called gingiva. The tissue that is holding your teeth is the gingiva. So this is called Entamoeba gingivalis. Leave the textbook and yes, we already talked about these, so just leave them. Pass them, yes. Also these, pass them, online references. We are still in chapter 3, amoebas. Taxonomically speaking, we are still in the Sarcomastigophora, Sarcodina, amoebas, Entamoeba gingivalis. In the site of infection, we are still in the intestinal lumen, because from the mouth to the anus, this is all called GI. So it's here. So it's still considered GI tract parasite, but it's all the time in the mouth. So it's still intestinal lumen. Entamoeba gingivalis, the oral amoebiasis, so it's the one that is in the mouth, oral, and this is the life cycle. Don't be surprised that this Entamoeba species does not have cyst. No cyst stage, only trough stage. All the time we kept saying cyst and trough, cyst and trough. You remember in the others? Histolytica, Hartmanni, Moshkovsky, Dispar, Indolimax nana, all of these, cyst and trough. This one, only trough, no cyst. Therefore, the method of transmitting this parasite is going to be a little bit different. Since we don't have a cyst, and the cyst is usually very hard and can resist the digestion in the stomach, and therefore it doesn't go down. Because what happens if this trough goes down to the stomach? It's going to be digested by the stomach acids. Good for this parasite that it doesn't go down. It's all the time in the mouth. Therefore, this is the trough is the infective stage and the diagnostic stage together. And how is it transmitted? It's transmitted by kissing mainly. Mouth-to-mouth kissing, using utensils. If you are using, let's say, the toothbrush of somebody else by mistake at home, also you may be using the same cup for drinking, utensils that go in the mouth. Therefore, this is the only method of or mode of transmission. It's non-invasive. It's only in the mouth and doesn't cause very much harm. This is the trophozoite. It has centric caryosome. It has peripheral chromatin as the other Entamoebas or Entamoeba species, and the chromatin distribution is even. And also, this is something which is peculiar about this. If you remember in the Entamoebas, we talked about one species which has red blood cell in the trophozoite. Which one? Histolytica. Very good. This one also has the same. It has RBCs plus white BCs, white blood cells, leukocytes. So now we have two Entamoeba species that might have cells inside the cytoplasm. Entamoeba histolytica and gingivalis. But the histolytica in the GI, in the intestine, and this one in the oral cavity, the mouth. Therefore, they call this oral amoebiasis. Fine granular cytoplasm, erythrocytes and leukocytes, multiple pseudopods, ingested food particles, So they eat because they are in the mouth, and in the mouth lots of food. So when they go around, they keep eating. So you might find food particles inside. Multiple pseudopods, yes. False leg. Size is 10 to 20. What you see here is the drawing and the figure. And what you see here is the reality stained by trichrome stain. Little bit of a difference. As I told you all the time, reality is different. So you see here the nucleus, you see some cells inside. No cystic stage, as I told you. Active motility, and I will show you what active motility means. Of course, when I say active motility in amoebas, all amoebas are slow, but one is faster than the other. This one, like E. histolytica, is very fast relatively, and this one active motility. And we have a video here. Single nucleus, as in the case of all amoeba trophozoites, they're all single nucleus. Microscopy. Wet mount. Do we use iodine here? No, we don't use iodine. Why? Because we don't have cyst stage. And usually iodine stains the cyst only. The trough, it kills it. Therefore, we do wet mount here, but using saline only. PCR. Stain smear, you can do trichrome stain, as in the case of all parasites. Samples. In the intestinal samples, our sample is stool, feces. Here, because it's in the mouth, the sample is something different. What is it? Mouth scrapings, so you can scrap between your teeth. Gingival areas here, around the gingiva, between teeth. That's one. Tonsillar crypts in the tonsils in the back. Pulmonary abscess, it can reach down to the lungs in some cases. Sputum, so it can be in the mouth, and sometimes, if you are lucky, it can go down to your lungs. And plus, it has been also isolated from the vagina. So vaginal and cervical material. So in the mouth and the vagina. Few cases or rare cases. Because it's rare, they call it oral amoebiasis, not vaginal amoebiasis. If you see here, look, here we have single nucleus, blue arrow, and next to the blue arrow, we have a small bilooped. You see here, it looks like loops of neutrophil. So this is most probably, it's white blood cells. Most probably in the middle here. Chromatin is even, difficult to see, I know. But white blood cells, you can see it. This is not red blood cell because it has loops. Hematology, you have the neutrophil with loops, multi-loops, minimum 3 in hematology. Red arrow, that's the white blood cell. This is much better here. You can see the next one, the second image, you can see the nucleus, and in the middle you can see the dot, the karyosome, and little bit of a chromatin around, uniformly distributed or evenly distributed. And then these black dots here, patches, these are the cells. White and RBCs. Sometimes you cannot tell which is which, but if you have loops, then this is white. White, see? White or RBCs. We cannot tell because the loops are not visible. Then we have a third picture, which is trichrome also. Look here. You have to have a good observation here that this is the nucleus. See this one? This is the nucleus with the chromatin and dot in the middle. And these here are white blood cells and red blood cells. So if I ask you, which Entamoeba has white and red? Gingivalis. Which Entamoeba has red? Histolytica. So these things you have to have in mind. Karyosome, centric, peripheral chromatin even, white blood cells, the red arrows here. This is a video which we took from other sources, and it shows you here the Entamoeba gingivalis moving with bacteria around. So you can look here. Because this is saline, unstained, difficult to see where the nucleus is. But if you look here, there are pseudopods. Mode of transmission. As we said, the mode of transmission is kissing, mouth to mouth, droplets via drinking utensils, or using somebody else's utensils like toothbrush and others. So if you put them in your mouth, then there's a problem. Reported infrequently, which means rare, in vaginal and cervical smears from women who use intrauterine contraceptives or devices. We call them IUDs. These are the devices that the women insert in their vagina so as not to have babies. We call them IUDs, intrauterine device. It's like a T-shaped, letter T. So it has been found that women who use this, they had Entamoeba gingivalis, but not in all cases, rare cases. So don't have in mind that every woman using IUD will get this. It's rare cases again. Oral amoebiasis, symptoms, asymptomatic, so you can have it in your mouth for years without you noticing. Recovered from patients with pyorrhea alveolaris, where you have pussy patches inside your lung. Associated with dental caries. You know what dental caries is? Pulpitis, gangrene, and stomatitis, and other infections in the mouth. So when you have poor hygiene in your mouth, most probably this is going to be there. So how do we prevent this infection? Simply, you have to have good mouth hygiene. Brushing teeth daily, good oral hygiene. And if the lady has this parasite, Entamoeba gingivalis, then she had to remove the IUD. Treatment, no drugs. The only thing you have to do is to remove the underlying cause, the original cause. A very bad oral hygiene, just clean your mouth by regular toothbrushing. This is the table which all the time we looked at. So we have gingivalis. So it's the only Entamoeba species that have no cyst. Granulation in the cytoplasm, it's fine. Centric, even chromatin. Chromatoid bars, no. Yes for the red blood cells plus white blood cell. So if you look here, you will see that this is the only one that has red blood cells plus E. histolytica. But there are differences in the site of infection. This is in the mouth and this is in the intestine. Plus white blood cells. They ingest bacteria. Rapid directional movement, yes, like the histolytica, and vacuole cytoplasm, yes, large. So this is the idea. This is the differential morphologic characteristics of all the species of Entamoeba. The end with this. By finishing this, we finish with the GI tract amoebae. Now we go to the amoebas that are where? In the brain. We have 3 brain amoebas. And these brain amoebas are all the time pathogenic, and not only pathogenic, fatal. They have high case fatality rate, which means that if you have 10 people or 100 people, more than 90% of them will die, more. So in most cases, people die. Because by the time you discover them, it's too late. Let's see the first one, which is Naegleria fowleri. Chapter 3, amoebas. So if you look here, taxonomically speaking, this is the taxonomy flow chart in front of you. Have a look at this. It's still Sarcomastigophora. Still we are talking about Sarcodina. And still we are talking about amoebas. So Sarcodina, Sarcomastigophora, Sarcodina, amoebas. Still we are in the amoebas. In the site of infection flow chart, you can see that we are not in the intestine, we are in the brain. Amoebae, Naegleria, Naegleria fowleri. It's good all the time to have these two flow charts every time we start a new chapter, or even every time we start a new parasite. Because these two flow charts, they tell you where you are, so you don't get lost. We have many parasites and students usually get lost easily. Naegleria fowleri, pathogenic free-living amoeba. What does this mean? It's pathogenic. We know what pathogenic means. Free-living amoeba. This amoeba doesn't need the humans. It can live in the environment alone. In the ponds, water sources, in the soil, it can live alone. Doesn't need the human. So that's why we call it free-living. And humans are just incidental host. They come by accident to this amoeba, or the amoeba comes to them by accident. Look at this. This is the NBC News. This is how media perceives the cases. Look at this. Brain-eating amoeba kills person who swam in North Carolina Mankind Lake. Look at the life cycle. There is something interesting about this amoeba, is that it has 3 stages. Every time we said that amoeba has 2 stages. Cyst, trough. Gingivalis has only trough. This one has cyst, trough, and there is a flagellated form. You know what flagella are? So it's a whip-like structure that helps the amoeba move. So look at the cyst. Number 1, that's a cyst. Look at the life cycle. Then we have a trough. So the cyst converts into trough. In the soil and the lakes, everything from cyst to trough, from trough to cyst, takes place in the environment, like water resources, like lakes. Then this trough sometimes converts into flagellated form. Look at this here down here. We have pear-like structure with the nucleus, karyosome in the middle here, and we have two flagella, so that can move very fast. So this is the only amoeba that has 3 stages. Even the second stage, which is the trough, it can also, by promitosis, it can convert to itself also. The trough and the flagellated form, these 2 forms, they are, or the infective stage. So you can either be infected by the trough itself or the flagellated form. But the cyst does not infect you. Understood? What is the mode of infection here? Well, while you are swimming in the pond, some of the water goes inside your nose. And this trophozoite, if it's in there and you have bad luck, it can get inside your nose, and through the olfactory nerve, it travels with the olfactory nerve to your brain and initiates infection. This is how you get the infection. Swimming, water inside your nose, and then it goes to your brain via olfactory nerve. And what does it cause there? Have a look. It causes a disease called PAM. What is PAM? Amoebic meningoencephalitis. For short, encephalitis. Abbreviation PAM, P-A-M. Trophozoite in CSF and brain tissue. So when they collect CSF from you, they make smear, they can see the trophozoite. Flagellated form occasionally in CSF, not very much. Usually they see the trophozoite. The question is that, what role does the cyst play in the infection? There is no role. Cyst does not play any role in the infection. Cyst is all the time in the environment. Doesn't come to you. In the GI tract amoebas, if you remember when we talked about this, all the time we said the infective stage is cyst. Here, cyst has no role. The infective stage is trophozoite or the flagellated form. Understood, all of you? This is how it looks. This is the Naegleria fowleri trophozoite. No peripheral chromatin. The karyosome is centric, which means in the middle. If you look at this image here in front of me, the trophozoite, you can see that it's not centric. It's eccentric. Have a look at it. In the book and in the literature, all the time the karyosome in the middle. For unknown reasons, maybe it's a mistake, they put the karyosome eccentric on one side. Did you notice this? Large centric karyosome, granular vacuoles, and they have blunt pseudopod. You know, like the E. coli. Pseudopod, the false leg, and it's blunt. And it's a slug-like. You know what slug is? The mollusk that is found near humid areas. Size 8 to 22, elongate, single nucleus as all the time. This is the cyst, centric, large karyosome, no peripheral chromatin, as you see here. A granular cytoplasm, thick cell wall, single nucleus here, in culture and environment, not in human. Why not in human? Because the cyst doesn't play any role in the infection of the human being. It's just in the environment. This is the third form, which is flagellated form. It's a pear-like shape, like a pear, and it has nucleus, karyosome in the middle, centric, and it has two flagella. Flagella for movement and motility. Size 7 to 15, smaller than the trophozoite. That's how it looks here, DIC microscopy, pear-shaped, jerky movement or spinning. So under the microscope, if you take CSF and this is there, you can see it moving jerky, shaking, and or spinning around itself. Now look at some of the slides taken from patients. So here in the middle, you can see the trophozoite here, and this one is a neutrophil, this one a neutrophil. So we have the trophozoite in the middle. That's Gimza stain. Leukocytes around. Of course, the leukocytes here are not inside the trophozoite. They are next to it, not inside it. Look at this. This here, blue area here, this is the trophozoite. And this is a neutrophil, this is a neutrophil, this is a neutrophil, another neutrophil, another neutrophil. So it looks that the patient has lots of neutrophils in the CSF, which means this is abnormal. It can indicate that this is bacterial infection, septic meningitis, because amoebic meningitis is very rare. And because of this, fatality rate is very high. Most of the patients die. The same here, the last one, the last picture here. Look at the neutrophils, too many neutrophils. These might be also monocytes, 1, 2, 3. These are monocytes. The multiloops here, if you look, the multiloops here, they are neutrophils. And the one in the middle here, the one in the middle where you see the red arrow here, this is the trophozoite of the Naegleria fowleri. And you can see in the middle, there is the karyosome, the nucleus with karyosome centric. Diagnosis. CSF wet mount. They take the CSF from the back of the patient and just wet mount under the microscope, or better, make stain. Gimza stain, trichrome stain, and you will see the trophozoite alone mainly. The pear-shaped, it can be seen, but not like the frequency of the trophozoite. Do we see cyst here? No, because cyst does not play any role in the infection. Cyst is in the environment. Culture, media and presence of bacteria like Escherichia coli, this is just for research. Serology, molecular biology, PCR. Epidemiology. In warm areas, so this is a very general word, the knowledge about this is scarce. In lakes, ponds, poorly chlorinated swimming pools, streams, small rivers, in soil where there is thermal discharge of power plants and geothermal wells. It looks that it likes heat and electrical fields. Looks so. Chain of infection, which we call mode of transmission, entering of contaminated water inside your nose while you are swimming. And inhalation of dust. This here, the two modes of transmission, the first one is the most encountered. The second one, inhalation of dust, because of the lack of real knowledge about this parasite. Symptoms: PAM, meningoencephalitis, stiff neck, your neck is really stiff, smell, taste alterations, sometimes you lose your taste and you lose your smell, you cannot smell, you cannot taste, that's at the beginning. Then blocked nose, your nose is all the time blocked with mucus, lots of mucus, we call it stuffed nose or blocked nose. Seizures. Koenig's sign. What is Koenig's sign? Let me show you Koenig's sign. This is Koenig's sign. Koenig's sign is a maneuver used by doctors in hospitals where you have the patient lies supine on his or her back, and then when you move the leg 90%, knee cannot be fully extended. If it's extended, then you are going to have a bit pain in your neck. Okay, so we have Koenig's sign, we have sore throat, you have nausea, little bit dizzy, nausea, and look, very important, very, very important, death within 3 to 6 days. So all of these symptoms, they appear at the beginning. But the problem with this parasite is that once it's in your brain, it will not give you time because it eats your brain. And because of this, fatality rate is high. Why? Because of these signs, because it eats your brain, and most importantly, because the diagnosis is delayed. People will not think about amoeba in the brain. So lots of time is lost while they are looking for the cause. And by the time they know it, it's too late for you or for us. So death within 3 to 6 days of the onset of symptoms. Once you get these symptoms and these symptoms become severe, then the death is inevitable and in a quick time. Therefore, it has high fatality rate. Prevention. How do we prevent this? We lack full understanding about this parasite. Therefore, we think that you should have good chlorination of swimming pools. Swimming pools, they should be chlorinated properly. That's one. Prompt reparation of cracks in pools. If you have in the pool lots of cracks in the walls of the pool, you have to repair them, renovate them. And most importantly, health education and awareness. But as I told you, still there is no full understanding of this parasite because it's rare. What is the treatment? Until now, there is no effective treatment. Can you imagine? No effective treatment. But those patients who had this disease, they have been given cocktail of drugs. Example, amphotericin B, intravenous. Amphotericin B is drug for fungus and also can be used for visceral leishmaniasis. Just blindly, hoping that it will work. They have been giving meconazole, another antifungal, intrathecally inside the spinal cord. Not in the spinal cord, but in the spaces between in the spinal cord where there is fluid, intrathecally. Plus rifampin, intravenously. So they have been given different treatments, different cocktails of treatments, hoping to help the patient. Sometimes it worked, sometimes it did not. That's the end. Let's go for the other one. Let's talk about Balamuthia. So let's continue. These are the textbooks. Let's just move. So again, this is the taxonomy flow chart. Still we are in amoebas, Sarcomastigophora and Sarcodina. Then to the sites of infection flow chart. This one, we are in the brain area, amoebas, Balamuthia. Okay, so it's Balamuthia mandrillaris. Again, Balamuthia mandrillaris. It's pathogenic. Whatever goes in your brain is a problem. And it's a free-living parasite. And this means that this parasite doesn't need the human beings. It's all the time in the environment. If this parasite comes to you by accident or you expose yourself to this parasite, then the infection occurs. Therefore, we are just incidental host or accidental host. Balamuthia mandrillaris. This is so we have here the cyst, and the cyst converts into trophozoite, and then the trophozoite converts back into cyst. And all of this process is happening in the environment, in the water, in the soil, in the environment, yes? And even the trophozoite can multiply by mitosis. You remember when we talked about the GI tract amoebas, all the time we said that they multiply by binary fission. 1 comes 2. But here we have mitosis. Then what happens? This cyst and trophozoite, so we have only 2 stages here, not like the Naegleria fowleri. Here we have 2 stages, like the GI tract amoebas. Cyst and trophozoite, they can get access to you. How? Either through the nasal passages, and then they go to the lungs, or sometimes through the wound. If you have a wound in your skin, in your hand, in your leg, then this amoeba can go inside the wound. And not like Naegleria fowleri, the trophozoite and the flagellated form. Does the cyst play a role there? No. Here, cyst and trophozoite can go inside the wound or can go inside your nose and go inside the lung and cause infection. And what do they cause? Again, encephalitis. This is amoebic encephalitis or granulomatous amoebic encephalitis, GAE. Cyst and trophozoite can be found in the tissue. So both of them are infective and both of them are. If you look here, this is the Balamuthia trophozoite. It has large centric caryosome. Even because it's very large, the caryosome, some people they call it a nucleolus. Not a nucleus, but small. Nucleolus is a small nucleus. It has a filamentous pseudopod like tree. You can look at the image here. No peripheral chromatin. Size 15 to 60. You remember the Entamoeba coli? Very big. This is like that one. But that one, Entamoeba coli, it doesn't have filaments like tree branches. It's only very big and that's all. This one has branches like tree. We call them filamentous pseudopods. Single nucleus. Rarely it has 2. The caryosome, sometimes it can be 2. Not only big 1, 2, but rare. Even sometimes it can be 3, but in trophozoite, inside the tissue. And in the tissue, by the way, this filamentous shape, it disappears. It appears like Entamoeba coli. This filamentous shape is only in culture or in the environment. But when it's in your brain, I will show you. Look, here, we have it. Look here. The image, the figure, filamentous, branches like tree. Here we don't have branches. If you look, it's just like Entamoeba coli. So the filaments are in the tissue, they disappear. It looks so from what we are seeing. This is the cyst. We have large karyosome, exactly as in the case of the trophozoite, no chromatin, and the cell wall has 3 layers, triple cell wall. Of these three layers, only 2 are visible. And the outer one has wrinkles. You know, when we get old, our skin becomes wrinkled, decoration or cortication around. That's the outer one. So this is a diagnostic feature. When you see the wrinklation around, then this is diagnostic feature. No peripheral chromatin, and this is the image. And below the image, you see the original one. See? These are the wrinkles. And this is the nucleus. Looks so. 10 to 30, the cyst, which is big. Single nucleus, wrinkled outer cell wall, as seen in the hematoxylin and eosine stain, or see as seen in the diagram, the figure. Microscopy is the main one. Culture, serology, not very much, like media in presence of primate hepatocytes, liver cells. These are for research. IFA, immunofluorescence assay, molecular PCR from CSF or from brain tissue. In the microscopy, you have to stain, either by Giemsa, trichrome, hematoxylin, eosine, H&E stain, any will do. Giemsa is very simple and very fast. And you can see the trophozoite, and the cyst. Brain tissue biopsy, rarely can we see it in CSF or skin or lungs or eyes. This is very rare, but mainly in the brain tissue, where they take biopsy. Look, here we have a cyst, and we have the wrinkles around. Blue arrows here, cytoplasm granulated. We have here 2, all of them, cyst, brain tissue, cyst, brain tissue. Wrinkled outer cell wall, this here. Cytoplasmic granulation, this here. Binucleated, this is one nucleus, and this is a second one. Again, brain tissue, hematoxylin and eosine stain, and the wrinkles are seen here. Cytoplasmic granulations, but we cannot see the nucleus here. Difficult to tell. Not like here. We have 2. This is Balamuthia trophozoite. Phase contrast microscope, single nucleus here in the middle, and filamentous pseudopods. See, these are the filaments. Looks like a lizard or any reptile. But these filamentous pseudopods, we don't see them in a human infection. In a human infection, it becomes just like any other ordinary trophozoite. So here we have the trophozoite, and we have here maybe white blood cells, and we have in the middle the nucleus with large karyosome, and what compared to the filamentous form, which is usually seen in culture or maybe in environment. Cytoplasm granulated here. That's a big one. Here this is a trough, single nucleus, yes, but in the middle we have binucleoli or bikaryosomes. They call them nucleoli more than karyosome. So multiple nucleoli, that's 1 and 2, very clear. We see here like we have a cytoplasmic, we have granulations, we have vacuole here. Balamuthia here begins as a skin lesion. So if it gets access to our bodies via open wounds, then begins as a skin lesion. Then goes to the brain and causes GAI, granulomatous amoebic encephalitis. Fatality rate 98%. With amoebic encephalitis caused by Balamuthia mandrillaris, most of them die. Double vision, because it affects the brain, so you see things too. Difficulty in swallowing, because it's affecting the central nervous system. Seizures, Facial paralysis like this. There is paralysis in the face, so you cannot move your mouth. Just like if you have palsy, cerebral palsy. Nausea, paralysis, vomiting. All of these, difficulty swallowing, double vision, facial paralysis, seizures, paralysis, they indicate that something wrong is going on with your brain because of something, and it tends to be Balamuthia. And the treatment, as I told you, there is no, well-established treatment for this. But people who had Balamuthia, they were given these drugs. Miltefosine, oral miltefosine, fluconazole, oral, this is also antifungal, pentamidine. So all of these, or cocktails of them, have been given to the patients in an attempt to save them. Sometimes it works, and most of the time does not. Look, the erythromycin, antibiotic, flucytosine, sulfadiazine, all of these have been given. Mode of transmission. How does Balamuthia get access to our bodies? But if you are gardening, working in the garden, playing with soil and dirt like children, inhalation or through wound. These are proposed modes of transmission. Cyst in soil and dust, usually. Epidemiology, Latin America, Peru, Asia, Europe, and Australia. These here are countries or continents where Balamuthia has been reported. Prevention, undetermined, unclear. See, because it's rare, no drugs, no full understanding. Therefore, you cannot tell people what to do in order to prevent this. So this is it for the Balamuthia. Let's go for the third and last one, which is Acanthamoeba. So Acanthamoeba is here. Taxonomically speaking, we are still in Sarcomastigophora, Sarcodina, and amoebas. Acanthamoeba. If we talk about the sites of infection, we are in the brain. Acanthamoeba. Acanthamoeba, it's just like the previous two in the brain. Pathogenic all the time, free-living amoeba. Usually its life cycle is complete outside the human, and humans are just incidental hosts. This is the life cycle. As you see here, we have these are the cysts, and we have exactly as the Balamuthia. We have here decorations and wrinkles around. Okay, so the cyst here matures and converts to a trophozoite, and the trophozoite back to cyst, and so on. This is the life cycle, and it takes place in the environment. Look, even the trophozoite, by mitosis, it can reproduce and multiply into other trophozoites. So this trophozoite gives rise to more trophozoites via mitosis. The cyst and the trophozoite, exactly like the Balamuthia, both of them, cyst and trophozoite, they can enter our body through eyes. They can go through our eyes if we are swimming, maybe, or if we have contaminated eye drops. They can get in our eyes and cause problems. Or they can get through nasal passages when you smell, and then from the nasal passages, your nose, nasal cavity, the nares, and then it goes down and to our lungs. If it's in our eyes, then it's going to cause this disease, keratitis, severe keratitis. If it gets access to our lungs, then it's going to be disseminated and it's going to cause the same thing as in Balamuthia, granulomatous amoebic encephalitis, GAE. Sometimes also, like the Balamuthia, it can go through our wounds. If we have wounds on our bodies, then, and the wound is open, not sterilized as should be, then it can get access through the wound. Again, this is like Balamuthia, that the cyst and the trophozoite are infective stages, not like Naegleria, only the trophozoite and the flagellated form. So cyst and troph, infective, cyst and troph, diagnostic. This is how it looks. Large karyosome. It looks a little bit eccentric here. Although they say sometimes it's eccentric. No peripheral chromatin, as you see here. And you see granular vacuoles in the cytoplasm. And most important, the Acanthamoeba. These are the pseudopods, and they look like spines, like needles and spines. That's why they call it acantha. Pseudopod, it's a pseudopod that looks like spines. This is how it looks. 8 to 22, not very big. Not like the Balamuthia, which was almost can reach up to 60 micrometers. Trophozoite, this is the trophozoite phase contrast. See how it looks? It looks like something like a reptile or something. Sluggish motility, not very fast. Lots of vacuoles, if you see here. Vacuoles, granular vacuoles, cytoplasm. Single nucleus, this one here, but you cannot see the nucleus here because it's not stained. Usually nuclei, they are not visible. Like mostly they are DNA, and DNA is colorless. And this is the cyst of the Acanthamoeba. Centric, karyosome, and large, no chromatin, or sometimes a little bit, but no chromatin in general. Granular disorganized vacuoles, not seen in the diagram, but maybe in the images we will see them. Double cell wall, so we don't have triple cell wall like the Balamuthia. Balamuthia has triple cell wall, and two of these three are viewed, and the outer one is wrinkled. This one, the outer one, we have two here, and the outer one is rugged. Sometimes it's jagged. Both are the same. Size 8 to 25 micrometers. Diagnosis. So we talk about CSF here and corneal scrapings from the eye, because sometimes it gets access to our eyes and causes keratitis, or CSF because it goes to our brain and causes GAE. Stained smears of CSF and corneal scraping, so you can do it with mount or you can do stained smears. Gimza, easy and very quick, or trichrome, lengthy and demanding. You will see trough and cyst. This is how it looks. This is a trough from the CSF. If you see here, large nucleus, karyosome in the middle, little bit eccentric. And at the end here, you can see spiny Acanthamoeba. Here, the red arrow here. This here in a tissue. These are the cells around, and in between here, you have the trophozoite, and we have the nucleus in the middle with large karyosome, but no chromatin. It's even better here. You can see that this is trichrome stain again. Cytoplasm is granulated. This is cytoplasm. You can see like a vacuum here, but these are the cell walls, and the outer one is rugged or jagged, as you like. This is the IC Nomarski microscopy, spiny Acanthamoeba, and this is the nucleus. Single nucleus. This is trophozoite from corneal scrapings in the eye, and this is the trophozoite with large nucleus and large caryosome within. Vacuoles, you can see vacuoles here. If you look here, Acanthapodia is absent. It looks that this Acanthapodia is not something regularly seen. Look here, cyst in a tissue. This is tissue, brain tissue. That's a cyst. That's a cyst. We have two here. And you have the jagged or rugged cell wall viewed easily. Symptoms of the Acanthapodia infection: stiff neck where the neck difficult to move, headache, seizures, vomiting, nausea. This is GAE, encephalitis. If the infection is keratitis, Acanthamoeba keratitis, aka, then this is the eye. See, the eye is totally destroyed. You lose sight, loss of vision, ocular pain. I took this from one publication, I think, where we have Acanthapodia species, many species, 10 species of Acanthapodia. Some of them cause GAE, some of them cause KA, and some, like these two here, cause both. And this one here cause both of them. Acanthamoeba, it's worldwide. It's not restricted in any geography or any continent, maybe because of the lack of full understanding. AK, Acanthamoeba keratitis, contact lens wearers. Some people, they don't like to wear glasses, so they use the alternative, contact lenses. And they have a washing solution for these contact lenses. This washing solution for the contact lenses, it can be contaminated. It can be saline you make at home. So be careful that this is one of the ways that Acanthamoeba can cause keratitis, AK. Chain of infection or mode of transmission, AK, contaminated contact lenses, as I said, and GAE, undetermined, not fully understood. So these three amoebas, brain amoebas, still they are mysterious. Acanthamoeba treatment, AK, keratitis and the eye infection, difficult to treat. Can you imagine? JIA, high fatality rate. People mostly die. And people in general, from these three brain amoebas, they die in high percentages. Why? Because mainly of delayed diagnosis. Most of the doctors, they think that, yes, brain problems, it's bacteria, meningitis, or viral meningitis, septic, aseptic. And by the time they find out that the patient is suffering from amoebic meningitis or amoebic encephalitis, the time is too late. The destruction and the damage has occurred. The drugs used for AK, although difficult to treat, but miltefosine, miconazole, rifampin, metronidazole. Yeah. And this metronidazole is the one we used for the GI tract amoebas, if you remember, the flagelle. So here we finish the chapter of amoebas, intestinal and brain amoebas.